How does blinding work in dementia clinical research? Imagine trying to figure out if a new treatment really helps with memory loss, or if people just think it does because they expect it to. That's where blinding comes in. Blinding is a method used in clinical trials to prevent bias, which can happen when people's expectations influence the results. In dementia research, this is especially important because outcomes often involve subjective assessments like memory tests or behavioral observations. In simple terms, blinding means keeping certain people involved in the study unaware of who is getting the real treatment and who is getting a placebo or control. This helps ensure that the results are based on the treatment's true effects, not on what people believe or hope will happen. There are different levels of blinding. Single blind means only the participants do not know their treatment. Double blind means both the participants and the researchers or clinicians assessing the outcomes are kept in the dark. Sometimes, a third level called triple blind is used, where even the data analysts don't know who received what until the study ends. To make blinding work, researchers use several strategies. They randomly assign participants to either the treatment group or the placebo group using computer-generated codes. The treatments and placebos are made to look identical in size, shape, taste, and packaging so that no one can tell them apart. The study drugs are often packaged in coded containers that don't reveal what's inside. When clinicians or evaluators assess the participants' cognitive or behavioral changes, they do so without knowing which treatment each person received. Sometimes, a separate team manages the treatment and can unblind only if there's a safety concern. Blinding helps reduce different types of bias. For example, it prevents evaluators from unconsciously rating someone's memory as better just because they think they're on the active drug. It also controls for the placebo effect, where people improve simply because they believe they are receiving treatment. Additionally, it stops caregivers or researchers from unintentionally influencing the participants' behavior or responses. However, blinding isn't always perfect. Some treatments might cause side effects that reveal who is on the active drug, which can lead to unblinding. For instance, if a medication causes a distinctive rash or imaging changes, it might give away the treatment group. Ethical considerations also come into play, especially if participants want to know their treatment after the trial ends. In dementia studies, double-blind, placebo-controlled designs are common because they help ensure that the observed effects are truly due to the treatment. For example, in trials testing drugs for agitation in Alzheimer's disease, researchers use identical tablets and codes to keep everyone blind. They also have blinded raters who assess the participants' symptoms every few weeks without knowing who is on the real medication. Overall, blinding is a vital part of clinical research because it helps produce trustworthy results. It involves carefully planned procedures to keep treatment assignments hidden from participants, caregivers, and evaluators, with safeguards for safety if unblinding is necessary. This process is essential for advancing our understanding of effective treatments for dementia and memory loss, ensuring that the evidence we're relying on is accurate and unbiased. Always remember, if you are considering participating in a trial or have questions about treatments, consult healthcare professionals for personalized advice.